Okay, wonderful. Thank you, Rob, for the kind introduction and for everybody who's joined us today on this webinar. Hopefully everybody can hear me clearly and also see my first slide. Okay, so what I'd like to do is provide a brief introduction to cell signaling, explain why antibody validation is important, and then I'll walk you through our strategies for IHC validation, and then how these antibodies are being used as a foundation for other assays and platforms. And then finally, I'll end with a few of our recently released products. So a little bit about cell signaling technology. We're a private family-owned company with a global presence, now approximately 500 employees. We celebrated our 20-year anniversary last year, and we're a company that's been founded by scientists that are still very active in research. Our focus is very much primarily application driven with regard to antibody validation, quality control, and also technical support. And throughout the last 20 years, we've developed 3 generations of rabbit monoclonal technologies in-house. Over 95% of our antibodies are developed in-house, and 100% of our manufacturing and quality control is also done in-house. And this is where our headquarters is based in Danvers, Beverly in Massachusetts, the USA. So we have complete control over the supply of all of our antibody products. Our portfolio today continues to grow. We now have over 7,000 products. And in addition to primary antibodies, we also have a number of sampler kits, cellular assay kits, ELISA kits, and proteomics solutions. And we can see here that the applications that we validate our antibodies for are ever increasing. So, for instance, our IHC validated antibodies, we have now over 1,000 products that have been validated for IHC. So why is antibody validation so important? I just wanted to make you aware, or maybe you are already aware and have seen some of these publications that I've captured here on this slide, that speak to the lack of reproducibility in science experiments. And it was actually quite alarming to see a couple of statistics pulled out here. Only 11% of previously published scientific findings can be confirmed. Only 28 of our in-house results match published data. And as you can see on the top right, or sorry, middle right here, we've got a publication here entitled Blame It on the Antibodies. And of course, as you can imagine, being from cell signaling, these are not publications that we'd like to see. And in fact, even as close as yesterday, there was a webinar on reproducibility that the ATCC hosted, and they polled 415 scientists, and 65% of those say that reproducibility is still very much an urgent problem. So the reproducibility crisis is really highlighting the many bad antibodies that are out there on the market. And the impact of this, of course, is a significant loss of time, wasted resources, and a lack of confidence in scientific findings. So CST continue to lead the way in how we validate our antibodies. We understand that using antibodies with high levels of specificity and lot-to-lot consistency is really critical to the success of science. And this is why we produce our antibodies in-house and spend a lot of time painstakingly validating our antibodies in multiple applications. So this slide demonstrates why it's extremely important that you use validated antibodies, especially when performing immunohistochemistry. Here we're comparing the IHC staining of a xenograft from 2 antibodies against phospho-MET. And both images you'll see show a specific signal for the protein of interest. But it's only then when you start to look at other applications, and in this case, Western blot, where we've looked at a number of cell lines which express receptor tyrosine kinases here. And it's clear that our competitive antibody in this particular case doesn't have, it's not specific at all. In fact, the level of noise raises concerns, about the non-specificity of the signal seen then in the IHC on the left. And so this demonstrates the importance of validation. So how do we do our validation at cell signaling technology? Well, we're really seeking to address two questions. Does the antibody stain where it's supposed to? And is there no stain where no expression is expected? And so for many targets, the biology is poorly understood and the expression pattern is mostly unknown. And then with many different cell types in each of the FFPE tissue sections, it's often nearly impossible to determine where staining should be present for more complex targets. So what we've done is we've standardized our initial validation strategy. We often start validation using cell pellet models. We generally identify many clones that stain as expected on cell pellet models, but then when we move to tissue models, many of these fail. Following cell pellet models, we'll move on to tissues. On one or small number of tissues with either a high or low or no expression expected. And then if the observed staining seems to be appropriate, our antibodies are then assessed on multiple tissue types, which is facilitated by the use of tissue microarrays. And then from that point on, we have a battery of tools that we use to further characterize antibody performance in FFPE tissues. So as mentioned, we don't do one assay. We do what's required to have the scientific confidence in the specificity of our antibodies. So here I've just depicted what our toolbox could look like, and one of the mantras at cell signaling is that no one single assay is sufficient to verify specificity of staining. So it's not enough that an antibody performs well in one experiment or on one high-level knockout cell model or on one positive tissue model. It's the use of multiple assays and strategies that help us to build the case for specificity. So what I'd like to do in the next few slides is I'll just show you some of the strategies that we use. So as I mentioned, we start off with FFPE cell pellets. These are fixed, processed, embedded, just like tissue samples. And they can serve as a useful first step in our validation. In this particular example, we can see that the clone performs well in IHC and also Western blot on cell lines of known positive and negative CD151 expression. And then we also look at cross-reactivity using cell pellets. So we can look at the cross-reactivity of an antibody against its related family members. And this is performed for protein targets that share high sequence homology with other proteins. In this particular case, we're using cell lines that have been transfected with related family members. So they are the EGFR, HER2 and HER4. And we can show that our anti-HER3 antibody detects just the HER3 protein and does not cross-react with the other tyrosine kinase family members. We'll then move on to tissues. And here's a good example of how we can use tissue samples with good internal negative controls, in this case cardiac muscle cells, to guide our validation. The antibody here is detecting smooth muscle actin, and in this section of mouse heart we can see that the antibody is staining the vascular smooth muscle as expected, while it's clean in cardiomyocytes. We can also leverage public available RNA expression data in the Human Protein Atlas to also help in our validation. And here we've got an example staining of CD39, which correlates well with high, low and very low tissues predicted by the RNA data. So running assays like this ensures that we can pick up the sensitivity that we require. Now, we not only develop human reactive antibodies at cell signaling, we also have an increased focus on making mouse reactive antibodies now that work in immunohistochemistry. We've generated a number of rodent models to aid in the validation of these targets. So as with our human antibodies, we'll test the mouse reactive antibodies on various normal mouse tissues and syngeneic tumour models. And in the example here, we can see that we're able to modulate protein expression using treatments, and the phospho-S6 expression has been inhibited upon rapamycin treatment. We'll also look at how orthogonal or non-antibody assays can be helped as well for our IHC validation. Examples here could be mass spectrometry or RNA scope. And here we've got mass spec data that's been performed on a cohort of lung cancers. And as we see, the staining with the antibody directed against the DLL3 correlates nicely with the protein expression levels detected by mass spec. And again, this gives us increased confidence in the specificity of an antibody. Another assay that we do is we often look at two different epitopes as well. And this is one of our best validation tools that we have at cell signaling. So in this example, we've got Notch2, where we expect to see membrane staining along with some nuclear signal representative of active Notch2, of Notch. But how can we be certain then that the distribution and level of staining is appropriate? And in this particular case, we've developed another clone that's directed against a different portion of the protein, also within the intracellular domain. And since both antibodies detect independent unique epitopes on the human Notch2, they show a similar staining pattern on multiple tissues and cell types, then we have increased confidence in the specificity of all the staining observed. We'll also look at species reactivity testing as well. So this is important to include if an antibody is predicted to react in additional species. So here we have two SHM2T2 antibodies that have been developed and show really nice staining in multiple human tissues. The staining's mitochondrial as expected. And since the antibodies are directed at different portions of the SHM2T2 protein, we can have confidence that the antibodies are good for these human models. But in this particular case, these antibodies are predicted also to react with mouse. And from Western blot testing and high sequence homology with the mouse SHM2 protein. But here, if you look, the antibody on the top shows nuclear signal in a subset of immune cells, while on the bottom we can see a very diffuse cytoplasmic signal that's observed in the pancreatic acinar cells. So note that both antibodies did not show similar non-specificity issues in humans, but it appears to be a mouse-specific, species-specific issue here. And therefore, we wouldn't recommend these antibodies for use on mouse, even though on Western blot and we had a high sequence homology with the mouse SHM2 protein. So then finally, another assay that we use when required is we can look at the co-localization of different targets. And here we're looking at the CD11C, which is a dendritic cell marker, and the XCR1 in red, which is expressed on a subset of these dendritic cells. So the co-localization of this stain helps us to verify that the XCR1 signal is on dendritic cells as expected. That was a very rapid tour of some of the IHC validation work that goes on, but as I mentioned, we do enough assays to give our scientists the confidence that we have the specificity and sensitivity required for a product to be commercialized. So moving on to how we might use these antibodies now as a foundation for other assays and platforms. We've certainly learnt that These can be used with autostainers, and on the left-hand side here we've got an example with the Leica Bond, which is the autostainer that we have in-house. And our IHC validated antibodies can also be used to develop dualplex and multiplex panels, and particularly used on multispectral imaging instrumentation, such as the instruments from Accoya, so the Vectra, the Polaris, and the Mantra platforms. First of all, just looking at the bond, our repertoire now of Leica Bond optimized protocols is approximately 125 targets that have been validated on the bond. This is the instrument that we have in-house. But all of our manual and bond protocols can all be found on our website. If there is a target that you're working on that isn't listed here, please don't hesitate to ask us. It might be on our to-do list, or it may just not be up on our website just yet. But when we do optimize our antibodies on the bond, we use our manual concentration dilution, optimal dilution first as a starting point, and then titrate up and down on the bond. And in this particular example with the PDL1, we can see that actually we can get equivalent staining with a higher dilution, 1 in 400 versus 1 in 200 on the bond itself, and still get equivalent staining. So what does this demonstrate? It demonstrates that our antibodies can be used on autostainers, so not just the Leica Bond, they're used on other autostainers as well, where the primary antibody incubation, is in the range of 30 to 60 minutes at room temperature, whereas our manual protocols would be have an overnight incubation at 4 degrees with our primary antibody. We've also developed assays using our IHC validated antibodies in a dualplex chromogenic manner. I'm not going to go into the details today in the interest of time, but we do have a couple of application notes. This first one here is on EMT, the epithelial mesenchymal transition, using E-cadherin and Vimentin. And it walks you through the optimization steps that we've used to develop a dualplex assay. And then the second example here I have is one that's been optimized on the Leica Bond, and this is using the FOXP3 and CD8 important T cell markers within the tumor microenvironment, and again walks you through the optimization steps that we've done to develop those dualplex assays. But in addition to our IHC antibodies being used on autostainers and also in dualplex assays, we're also seeing our antibodies now increasingly used in multiplexed IHC. And this is really primarily due to the increasing number of biomarkers that we have access to, and often the limited supply of biopsy material available, which is creating a need for multiplexed assays. And we're certainly seeing this is particularly attractive in the field of immuno-oncology, as we're able to maintain spatial context within the tumor microenvironment and know that that provides us with meaningful information. So we can look at co-localization of different biomarkers, we can look at the frequency and proximity of these biomarkers being studied. So all of our IHC validated antibodies in our portfolio are compatible with fluorescent multiplexed IHC, and so that's providing a large repertoire of targets that one can work with. So what do we need for multiplexed IHC? There's three key areas here. So if we're looking to run a multiplex assay, say a 4-plex or more, you're really looking for a specialized instrument, particularly, a multispectral imaging instrument is ideal, and that would be the Vectra or the Polaris or the Mantra, which will allow you to deal with multiple, enable you to work with different fluorophores and prevent spectral overlap, but it will also allow you to eliminate tissue autofluorescence. In addition to an instrument, we know that we need to work with some highly validated IHC reagents and antibodies that obviously work well in FFPEs, and also have a robust detection system. So here, this is where the OPAL technology and the TSA really comes into its own with multiplexing and signal amplification, and also works independent of antibody species or isotype. So at Cell Signaling, we are lucky. We work with a Mantra multispectral imaging instrument for our image acquisition, and then we're using the Inform Tissue Finder software for our analysis. And over the last 2 or 3 years, we've been able to develop several multiplexed IHC panels, some of which are captured here, predominantly in the IO space. And we can develop lovely, pretty images such as this. So this is one of our panels for immune checkpoints that's looking at a T-cell exhaustion panel where we're able to identify PD-L1 positive cells. We can identify, where we've got co-localization with CD8 positive T cells infiltrating the tumor. And of course, my colleagues at Akoya will go into a lot more detail as to how to get into, the tips and tricks of developing a beautiful imagery such as this. But I just have one slide here where I'll walk you through the process that we do at Cell Signaling. So we always make sure that we have a robust signal in a singleplex chromogenic assay for each of our different targets within our panel to start with, before we'll move on to optimizing then the antibody dilution used with a single fluorophore. We'll then look at a design of experiment, DOE experiment, where we'll look at fluorochrome and a pairing comparison, and we'll determine the optimum antibody and dye combinations. Brighter fluorophores are often paired with the lower expressing targets and vice versa. And then finally, we'll look to optimize the order of addition. And then, we end up with a nice table here where we've got our optimized conditions with our targets, our antibody dilutions, the fluorochrome used, and then the order of addition. And we've got a few application notes in this space now, but I'm just capturing this one here. But of course, my colleagues at Akoya will talk more about the tips and tricks of optimizing these types of assays. And then finally, another platform where we are seeing our antibodies being used now is on the Fluidigm mass cytometry Hyperion platform, predominantly for discovery work. And this is data that's been kindly generated by Noel de Miranda from the LUMC, where he's been studying colorectal cancer. And the images here are from one of his 20-plex panels, showcasing some of the CST antibodies that he's using. A whistle-stop tour, I know. Some of our recently developed products, although we're adding antibodies all the time to our portfolio, we release products every couple of weeks, particularly in the fields of cell biology, immunology, neuroscience, epigenetics, and ELISAs. We've developed very recently a couple of new stains that we've added to our portfolio. One is Vivid Purple, which is particularly useful for anybody who's wanting to study melanoma. And then finally, we also have a new Vibrant Red alkaline phosphatase substrate kit as well that we've recently launched, that's been used in some of our recent application notes, particularly our dual-plex assays. And so before I hand off to Virginie, a quick summary then of my talk today is that our IHC validation should be tailored to each target. We have various validation tools that are employed to validate our targets, and no one assay is sufficient. We use multiple assays synergistically to demonstrate that we have the required specificity before we commercialize a product. And we know that highly validated antibodies are really crucial in multiplexed assays to produce reliable staining. And that will obviously enable you as a researcher to have more confidence in the discoveries that you're working on. So thank you very much. We'll be taking questions later, but I'd like to hand over to Virginie now, who will talk more about the OPAL technology.
Hi, everyone, and thank you again, Janet, for introducing TSA, and I could go faster on some of my slides then. So just to give you a quick overview of what I will talk about in my presentation today. I will first tell you a bit more about who we are. Indeed, you might have heard about, you might never have heard about Akoya before, since our company name is quite new. Although we carry some technologies that have been around for quite a while and have been really well demonstrated through hundreds of publications worldwide. And then I will introduce you the multiplexing solution we offer in order to look at several markers on the same tissue section. And more importantly, I will give you today some tips and tricks and maybe pitfalls to avoid that I could collect over some years of experience on the field in order to ease or at least to speed up your multiplex assay development. And at the end, if I still have time, I will give you a short introduction to multispectral imaging. Janet quickly touched base on that, but this imaging solution offers you to look at multiple dyes on the same FFPE tissue section with autofluorescence subtraction. So who are we as Akoya? So maybe you have heard more about PerkinElmer. So all the tissue imaging products, also called quantitative pathology solutions from PerkinElmer, spun off with the support of some American investors and got fused with another very innovative technology from Stanford University and more specifically from Gary Nolan Lab called Codex. And the two together were really complementary to form Akoya Biosciences. So the reason why we generated so much interest and support is mainly because we saw a huge gap in between what was currently available in terms of multiplexing in tissue. So one, at least before, we had to choose between a low plex just to determine one or two markers on a tissue section, or high plex technology, but this high plex technology had to be done on homogenized samples or very expensive and very low throughput. But with Akoya, we now provide solutions to do with two complementary product lines. So one called Codex from Stanford University, as I said earlier, would enable you to do up to 40 markers on the same tissue section with a single cell resolution, whole tissue imaging, and in a very flexible and affordable way. But what I'm going to focus my talk today is on what we call Phenoptics, which is these quantitative pathology solutions imported from PerkinElmer, which enables you to do what I would call a medium-high plexing, so you can do up to 8 markers plus TAPI. Also with a single cell resolution, it's based on immunofluorescence, and you can go up to a 40x resolution. Whole slide multispectral imaging, if you have, and with a very high throughput, in a very high throughput manner. So, multiplex solution. We're today the only provider of reagents, instrument, and software to really provide you a full solution to carry your assay from the staining to the imaging and data analysis. I will focus again most of my talk on the staining part, so on the reagents part, using OPAL, and also giving you some tips and tricks who are also more related to general immunohistochemistry and immunofluorescence. So if you're attending this webinar today, I guess it's probably because you've been facing a lot of challenges using immunofluorescence or trying to multiplex. And I worked in research before. I've done a lot of immunohistochemistry, so I can totally understand all those challenges. So for example, you have to you had at least to care about antibody species and carefully choose your secondary to avoid any cross-reactivity. You had to sometimes some umbrella effect or steric hindrance, so you couldn't look at colocalized markers. You had to sometimes struggle with very low expression of markers where you get really low signal and it's completely masked by the autofluorescence of your tissue. And then you end up with crazy exposure time, you photo bleach. It could be a nightmare. But with OPAL and TSA, which also Janet talked about during her presentation, so TSA, by the way, stands for Tyramide Signal Amplification, and it has been around for at least 2 decades as an amplification method. So you will find thousands of publications as used for fish or even for tissue as an amplification method. But what we leverage at PerkinElmer and now at HIER is also the fact that those little fluorescent molecules, those tyramides, are covalently bound to the targeted epitope. And I would say that's the take-home message. The fact that those tyramides are covalently bound to the epitope, we can remove the antibody complex, so we can completely strip the primary and secondary, and we can come fresh again on the tissue with another set of antibodies. And that would enable you to use the same species of primary if you want. And that's really important. If you want, you can use only rabbit primary antibodies in your assay, and you don't have to worry about cross-reactivity or anything. So you get an amplification and you get an easier way to multiplex and also to look at colocalized markers, because we also found that this deposition is very discrete. It's in close vicinity to the antibody complex. As you can see, it's catalyzed by HRP enzyme, and so that's why you will always see in OPAL assay the use of secondary HRP. So how does it work? In terms of workflow, I would say you will do, as usual, a deparaffinization step if you're working on FFPE tissue section. Then you will do antigen retrieval, as you usually do for FFPE. Then you will block, usually for 5-10 minutes. Then you will add your primary antibody and you will leave it for 30 minutes or an hour, depending on the affinity of the antibody. Then you will perform some washes, add your secondary HRP of choice or the one we provide in our kit. You can wash again, and then you will apply the dye. So we have, as of today, 8 different dyes, which would enable you to do 8 plex plus DAPI. And then at the end, you strip the antibody complex. Only the dye will remain on the epitope. And then, and that's how you can repeat this sequence of staining cycles as many as you have markers to reveal. As many times as you have marker to reveal. And at the end, you will end up with a DAPI counterstain and MONT in order to analyze your biomarkers. So, as you can imagine, I mean, of course, if you repeat the staining cycles manually, it could take you over 1 or 2 days, at least 2 days for 6 plex. But now we have a close collaboration with Leica and also with Roche Discovery Ultra to be able to fully automate this staining protocol overnight. So from deparaffinization to DAPI, everything is automated on the Leica Bond or Roche Discovery Ultra. And we have special kits for them. So we wanted to make it available to anyone, not even depending on the microscope you have. So we have 4-color kits, which are compatible with any standard microscope that you might carry in your lab. And we also provide now a 7-color kit and a 9-color kit and a mouse kit if you don't work on human tissue section. We also have now this new mouse OPAL kit, which enables you to work on mouse tissue. And so I will give you just over 2-3 slides, an overview, a quick look or a glance at what we have performed internally using mostly only cell signaling technology, because on mouse tissue, we found that they have the most exhaustive list of rabbit-raised antibodies for mouse epitopes. And so, yeah, this proof of concept has been done on breast cancer tumor. And as you can see here, we were able to look at those 6 markers plus DAPI in a very precise and very specific way. And I'm happy to share with you the clone we use, the order of addition, everything you might need to perform this kind of staining. Also, and I will go quickly through that part, to get you started with OPAL, if you're interested in human immune markers, we also offer what we call panel kits, and those panel kits are ready to use. They contain everything you need, from primary antibodies all the way to DAPI, all the buffers, antigen retrieval, everything you need except maybe the wash buffer to perform your assay and to finish, start to finish. And yeah, so we have one for lung and we have one for melanoma. Also, we have developed this proof of concept to go up to 8-plex. And again, I'm happy to help you with sharing information about the clone. We use the order of addition to really help you develop this kind of assay internally. So now comes the tips and tricks part, because what we really emphasize also with OPAL is to give you flexibility to be able to use your own antibodies and to work with your own panel kit, to develop your own panel. So we provide online on our website, or you can ask me, a lot of documentation, application note, assay development guide to really help you in developing your 4-plex, 5-plex, or 6-plex panel. But, you know, we can help you with the detection method and the staining. But as you know, if you have done some immunohistochemistry before, you have lots of factors and parameters to take into account. And I really like this publication from Gillian et al. Called Garbage In, Garbage Out, because whatever you do in terms of staining, if you have a crappy, sorry for the term, tissue, you're not going to be able to do something nice anyway. So you really have to think about how you treat your tissue even before thinking about staining. So first, of course, the epitope retrieval is quite crucial, but also the way you fix your tissue is quite important. So as you can see here, fixation and what you use for fixation, formalin, it has to be fresh, it has to be buffered, and also how long you apply it on your tissue really has an effect at the end on the staining. So here, if you compare on the right versus left, so right is when it's only 3 hours, and left it's when, no, reverse, left 3 hours, right 8 hours. You can see that the staining is much better and you have much better pattern. And same for the deparaffinization. You know, I know there are lots of methods now for quickly deparaffinize and de-wax your slides. I don't believe in miracle, especially in histology, and I feel deparaffinization in the oven overnight at 55, 60 degrees is the best way to at least get rid of most of the paraffin away, which doesn't, of course, replace the xylene and ethanol, but we really see a huge impact in deparaffinizing in the oven overnight, or at least 2 hours before xylene and ethanol. And of course, use good slides. I mean, your samples are very precious and expensive. I know glass slides can also be expensive, but really it would save you a lot of samples, and it would really prevent your tissue sample to come out. So I would really encourage you use the Superfrost Plus or Thermoslides. So other key components when you think about your immunohistochemistry assay. So I guess you're familiar with antigen retrieval. What do I choose? pH 6, pH 9. Sometimes the antibody provider are nice enough and give you this kind of information. I hope cell signaling does. And yeah, also the blocking, because not all the blockers are working fine for your tissue type or your antibodies. So you really have to think carefully about the antibody diluent you use and the blocking you have to do before. And there are, I would say, two kinds of blocking. You can quench the endogenous peroxidase, and so usually you use H2O2 to do that, or you use protein blockers, because you can also have non-specific binding due to charged protein, and you can block that easily with casein and BSA. Then the choice of antibody, and that's also crucial. Really think about something you have validated internally using DAB, or you can use the literature, and of course you can trust, trustworthy provider, such as, for example, cell signaling, because as Janet showed, they spend a lot of time validating their antibodies, and I think it's really important to really make sure those antibodies are well demonstrated and validated. And then, of course, the secondary. So in our kit, in our OPAL kit, we provide a secondary antibody, which is anti-mouse and anti-rabbit. That means you can only use it on mouse and rabbit antibodies. But if you use other type of primary antibody species or other tissue type and you cannot use our secondary, please feel free to use any secondary antibody you would like, as long as it's conjugated to HRP. And yes, all antibodies are not created equal, so you really have to test different antibodies if you're not satisfied with the results you get. Then, of course, chromogenic, fluorochrome, I would say it's kind of your choice, but if you want to quantify signal, if you want to look at co-localization, I would really encourage you to use immunofluorescence. I won't go through this nightmare, and Janet already went through it, so I skip this one. So, sorry. Yeah, in terms of first thing, when you validated your antibody with DAB, or at least you know from a colleague that it works really well. First thing we ask you to do is a slight titration. So not a big one. It won't require you a lot of time, just 1 day maximum. Just test from the DAB or the vendor's recommended dilution, 2 times more, 4 times more. You will see with OPAL, you get a very nice amplification, but it's so you will be tempted to dilute, but don't over dilute it as well, because if you have a low expressing tissue block or a less well treated tissue block, you might completely lose your marker. So if the antibody doesn't cause too much background, I will still keep a good antibody concentration. So I don't think I have to really teach you how to do a titration, but what you have to look at is really looking at comparing those different dilutions and really look not only at the signal intensity, but also the background you get around. So here, as you can see, on 1 to 400 is too diluted, but the 1 to 100 gives you some background you don't want. And with multispectral imaging and INFORM, we're able to look at the immunofluorescence image and translate it into hematoxylin DAB view. We call it the pathology view because pathologists really like this tool, where you can really see from your fluorescent image you get what you will get if it was DAB. So it really gives you this confidence or this comfort when you look at your image to compare it with what you have with DAB. If you don't have Inform, of course, you can also do it from looking at the pattern and make sure you get the same pattern as what you were getting with DAB. Yeah, and one other thing when you're putting together your multiplex assay. So, you know, I always get the question, does heat affect my tissue? And I would say with confidence, no, it doesn't. It doesn't affect your tissue. We worked on many different tissue types, but it could have an effect on certain epitopes. And I would say those epitopes are very rare, but it's something to look at. So the best way I have found to look at the effect of several heating steps or stripping steps on your epitope, on your targeted protein, is to work on 3 serial sections. So you have 3 tissue sections from the same block, if possible. The first one you're going to treat after 1 heating step. The second one you will treat adding your primary, secondary, and so on, after 3 stripping steps. And the third one after 6 stripping steps, so heating steps. And then you will compare the signal to background ratio. So you will look at the signal compared to the background, and you will see if it increases. If it increases, you know that this antibody can be placed at the end. If you have a drop in signal, or you start to have a staining very diluted, then you know that you have to place this antibody at the beginning of the sequence. I hope it makes sense to you. If it doesn't, please write me an email afterward, and I will be happy to go over this step again. So yeah, this actually I got that from Roche when because Roche also has done a study, and they showed actually that only 5% of the epitopes are fully retrieved after 1 epitope retrieval. So when you think of it, I'm not surprised why I see in 80% of the case actually an increase in signal after several retrieval steps. So as you can see here, that was done in the Discovery Ultra using the CC1, CC2, so it's pH 6 or pH 9, and looking at the PDL1 expression. And actually you see an increase, a quite huge increase in signal through those repeated heated steps. So just think about it. And also in the pattern, you have to look, the signal is one thing, but you also have to make sure the pattern is well conserved through those sequence. Often also I have the question, what do I use for stripping? And I would say we're very flexible as long as it works. So use whatever you have in the lab, but make sure you do the right control. If you have a water bath, fine, eat it at 98 degrees, 20 minutes. Make sure you don't burn yourself. It happened to me a couple of times and it hurts. But you can use steamer, pressure cooker. PT-Link from DACO is the module you can use just to perform stripping. And of course, microwave. But microwave, you have really good ones and you have really bad ones. And yeah, just consult me if you want to use a microwave, because it could be detrimental and you want to make sure you have a good grade microwave. Anyway, so in terms of stripping, you can use 2 types of controls. You can check, so you do a full staining cycle, then you strip, and then you add the other antibody complex plus, you strip, sorry, and then you add only the secondary and another OPAL dyes. So as you can see on the right, you do your staining with OPAL 520, you strip, and then you come with your secondary HRP, OPAL 620, and as you can see on the right image, it's not fully stripped because you can still see some leftover, some binding of OPAL 620, which means you still have some primary bond to your tissue. So That's one way. The other way is, for example, if it's a CD8, use a nuclear staining. If it's a nuclear staining, it shouldn't be colocalized with a membrane staining. So it's also another way to check that you don't have false colocalization due to incomplete stripping. The good thing is that you don't have to worry about the OPAL dyes. They're very stable. And so then you just have to pick the OPAL dyes depending on the abundance of the marker, the brightness, but you don't have to worry about how they behave through those heating steps. Detection system. So as I said, you have to think carefully about what do you use, chromogenic, immunofluorescence. I would say how many plex you want to do really also determine what detection method you will use. Of course, also the expression level. If you have a very low expressor, I would really encourage you use those TSA or amplification method. And of course, colocalization as well, because colocalization is quite, it's quite common. You know, if you look at CD3, CD8, PD1, you're already at 3 markers per cell. So as you can see here, we have done this proof of concept where we look at 4 different markers on the same tissue, on the same cell membrane. But yeah, you have to titrate to make sure you don't oversaturate your marker. But it's totally doable, and it's doable only because you don't have this steric hindrance created by the antibodies. And yes, this flexibility in the choice of dyes you use as well. So the take-home message, as a matter of time, I'm going to skip some slides or go very quickly through it. OPAL really brings you the flexibility you need, and I hope this presentation was also good to give you some tips about what to think about when you perform a multiplex assay. And most of all, pick your favorite antibodies, because you know we can see on the market some ready-to-use kit, but you know I think it's really also important that you're able, if you want to look to use the antibodies you want to use and trust in your lab. So quickly, because Urs Ziegler has a Vectra 3, and you know if you want to look at more than 4 markers on a same tissue section, you really need a specific kind of instrument. And we carry also at Akoya, as we see that it's a very important piece of the puzzle, multispectral imaging instrument. I won't go, because I don't have time, into too much details, but multispectral imaging enables you to look at every pixel through a spectrum you get, and then we use the software to deconvolute, to do some spectral unmixing, which really gives you extremely pure quantification of your fluorochrome. And we're also able to learn the autofluorescence from the tissue and to completely isolate it from the image and the quantification. The good thing is we do it in a very fast manner. So now with the Vectra Polaris, we can scan 1 by 1.5 centimeter tissue section at 20x with 7 colors plus autofluorescence subtraction in less than 10 minutes. So we're also kind of the fastest, I think, in terms of scanning. And best of all, we also provide software with the instrument. It comes with the instrument, and it enables you, as Janet showed also quickly, because they have it also at Cell Signaling, it enables you not only to do the spectral unmixing and to remove the autofluorescence, as you can see here, but it also enables you to do some tissue segmentation, cell segmentation and compartmentation, phenotyping based on machine learning, so it's super easy. I'm not a bioinformatician and I can use it, so everyone can use it. And scoring as well, each score depending on the intensity level you get. If you have any questions, I will stay at the end after Björn's presentation, but if you want to reach me, please write me at v.goubert@akoyabio.com. And now I will leave it to Björn.
I picked a few publications, one about the workflow that just came out, one about prostate cancer. I also picked an older one, which is about pancreatic cancer, because this is a very difficult type of cancer to analyze. Also about skin inflammation in a mouse model. Very interesting, also very recent transplantation publication about kidney transplantation. And then I give you a very short outlook what's possible maybe in neuroscience with it. So starting with the first publication that I chose here, this is from this year, from March, so it's just a month ago. And what I feel is very interesting also maybe for your work, you see the group here, this is a technical paper, a method paper. The group here chose very many immune markers, but also checkpoint markers and others. And you see they created 5 different panels here. Interesting maybe to remark is that in each panel they use CD3 and CD20 kind of as a landmark also for the data analysis later on. And finally, what they did with these panels was they imaged it, of course, and then they looked into the cell density here. And this they did on lymphoma, and lymphoma is not so easy because in the immuno-oncology field, it's very common to have these solid tumors, but in lymphomas, the frontiers between tumor and stroma are not so clear. So this is a little bit more difficult type to analyze. And here in this study, and that's why I picked it, I think it's quite neat. You see, they automated the whole workflow nicely. So they started with an H&E analysis on their tissue samples if they are good for this study. And then they run the multiplex staining, as Virginie explained to us, in the Bond RX. Then they use the Vectra 3, so the same that is located in Zurich at the moment, and did a pre-scanning here and annotated then the regions for spectral imaging. And the spectral imaging was then performed in 20x resolution. And then it went over to the Inform software that we provide, and they did several parts of the analysis, including then a binary phenotyping, which means they only looked for always one phenotype and nothing else. And then in the next step, combined also these phenotypes to figure out double or even triple positivities out of this phenotyping. So you see the whole workflow was automated in terms of staining, but also in terms of analysis later. So the next publication I would like to show you here is also very recent. It's from April, even from this month. And this is about prostate cancer and vaccination. You know, there's a high interest in immunotherapy and vaccination using our technology. And but very often, if you talk about vaccination, it is that the vaccine recognizes the surface of tumor cells. But in this study, interestingly, it was also shown that the vaccination also has an influence on the infiltration of immune cells into the cancer region. And you see now on the right-hand side that they used quite a extensive panel here with immune markers, including also tumor markers, cytokeratin, and cytotoxic T cells for CD8 and CD4. But they also look for proliferation, Ki67, and a checkpoint marker, PDL1 here. And what they finally focused on was on CD4 and CD8 cells that they nicely could identify. And but the tumor itself is not so easy to deal with, and this is shown on this picture here. So you see that they identified the core of the tumor, which is marked as CT, but also the invasive margin here, and finally the rest of the gland. And they calculated how many immune cells infiltrated into the tumor, into the invasive margin, and so on. And they could show here nicely that vaccination makes a higher infiltration into the core of the tumor, but also into the invasive margin here. This is an older publication here about tumor microenvironment in pancreatic cancer, and this was probably one of the earlier studies where they used TMAs actually in an automated way on the very challenging pancreatic tissue or research here. So interesting here is they went already with a Vectra 3 above our 7 colors. So they did 8 color stains here on human tissue samples, but they also used mouse tissue samples with 7 colors actually, and compared the outcome here. And why I picked this paper as well is because not only having a lot of markers in place plays a role, but also the analysis. And here they did, with our provided R scripts that we provide for free, they did a spatial analysis. And you see this in this graph here, where they looked for a cell density within a certain radius around tumor cells, and in this example here with T cells, and they see when there are high density of T cells or lower density of T cells, they could then clearly see that this is also a factor that shows higher survival of the patients, which means not only the total amount in a section of T cells is important, but also the spatial relation to the tumor cells is important. And this is where our technology comes in place. Here, this is from last year, an example from a group from Frankfurt. Actually, they did studies on skin here. And it's actually not so impressive in terms of how many markers they used. It's actually not so many, but what's quite obvious on this here is that you see on the left lower 2 pictures, you see this is actually not from the publication, this is from us here, but it illustrates nicely the advantage of multispectral imaging, because you see a skin section here with only 2 markers plus DAPI, but you see on the left side there are a lot of collagen fibers, a lot of autofluorescence, but on the right you see that this is completely removed from multispectral imaging, and you can clearly see now the staining, and exactly this was also used in the publication here. So all these images, they had to remove a lot of autofluorescence to get the clear signal. And also on top, you see now that, as Virginie mentioned, we can now convert this imagery into a virtual pathology view so that you can interpret your data much easier. And now we are very proud of this publication here that came out, or is just coming out now in April, in the American Journal of Transplantation as a title publication here. And this is about the transplantation. And also here, people wanted to see what is going on, how is the immune system reacting on transplanted organs, on tissue. And for this, they had to use also several markers here, immune markers, but also markers for NK cells and for blood vessels. And then they measured if they determined the intra and extravascular location of those immune cells, and especially macrophages and T cells here. And interesting also to mention is the NK46 antibody here, or marker, because normally for NK cells you use a CD56, but this group also showed that the NK46 is much more precise and has a better outcome here. And finally, what they show with their study is that, or with this technique, they could really show that intravascular or extravascular, there is indeed a difference here. And I can give you some more explanations maybe after the talk, if you like. And the last thing is the outlook here for the neuroscience. You see now a brain tissue, very often you need quite big parts, big tissue pieces to look at, but brain tissue is highly autofluorescent as well here. But also for brain, the Polaris scanner is really nicely to work with, because it can remove autofluorescence and enables you to really show different kind of cells in high precision. And on the left lower graph, you see now that the signal to noise ratio increases extremely nice, especially for the bluish greenish dyes. As you probably know, there's less autofluorescence than in the reddish, or the more you go into red. So the increase is not as much, but for the bluish greenish, it's an enormous help if you can remove this autofluorescence. And that's already it. I just wanted to give you these teasers here, and show you how people work with this technology also in different application fields. And I think I could show you that the whole workflow can be more or less automated by now. You can see it's in different fields where people work with this technology now. You have many ways to visualize your data nicely, also with this pathology view, for example. And it's getting more and more in this high-dimensional data now, with more multiplexing, bigger tissue parts, and faster scanning. Because if we can scan slides in less than 10 minutes, then you can really plan high-throughput experiments with this technology. And with this, I would end now and show you again our email addresses, and hand over to Robert.
Okay, thank you very much to all the speakers. This session is now open for questions. There is already one question from Urt Ziegler. This is directed to Virginie. So we'll start off with that. How much does the signal diffuse during the HRP signal generation?
Can you repeat the question?
Yeah, the question is, how much does the signal diffuse during the HRP signal generation?
Oh, okay, in terms of...
I think how much does the signal spread? How localized it is, I assume.
Yes. So we have this publication who was performed using electronic microscope, and they were using nano gold. And from this publication, we can think that the deposition is in a distance, in a diameter of about 1 to 2 nanometer. I can send this publication to you, but you could see that before and after deposition with TSA using this nano gold, the size of the dots were not really increased, but the intensity was really increased. And so we believe, and also looking at really colocalized markers, that it really stays in a diameter of 1 nanometer around the antibody complex. The reason why is this chemical reaction with TSA is very fast. HRP turns those thymines into highly reactive free radicals, and if they don't quickly react with the tyrosine around them, they create dimers, and those dimers are washed away. So that's why they don't float around and, you know, spread too far away. I hope that answers your question, Urs.
Perfect. Thank you.
Yep. You're welcome.
So, anybody else? Some questions? We still have some time. There's another question from Josea Marie Mateos. Maybe I missed it, but is it possible to use this technology in cryostate sections? Good question.
So you haven't missed anything. I haven't mentioned it, and it's a very good question. Most of our experience with OPAL has been on FFPE tissue section, so formalin fixed paraffin embedded tissue section. But we also have users, we've heard users using it on frozen tissue section, but you have to be more careful, even more careful about how you handle your tissue. And I will highly recommend that before you start performing a staining cycle, that you perform a fixation step using neutral buffered formalin, so NBF at 10%. You stick your slide in the jar for about 20 minutes, and then it should really enable your tissue slide to be fixed onto the glass slide. Of course, since you've used the fixation step, you will have to do a quick epitope retrieval step to be able to work on to perform your staining and to reach the epitope. The other thing is to use poly-L-lysine coated glass slide, and also if you can avoid too many heating steps, it's recommended, you know, to do maybe 2, 3 maximum, and you can do H2O2 to quench the HRP instead of performing some stripping step. But I would be happy to take your question also offline to really discuss it in further details. But yes, you can perform OPAL amplification on frozen tissue section. You're welcome.
So, anybody else? Okay, so let me finalize with, I think we should close down, right?
Yes.
So if there are any more questions, you know, you have all our email addresses by now, and we will also, and we will also follow up. We will send you a link to this recording, and there in this mail, we will also mention the individual email addresses of the speakers. And so if any question pops up later in time, then you're free to send us an email with your questions. And with that, I would like to thank you all for your interest and presence, and we hope you found this useful. And especially we hope that all of you and your family and friends stay safe in these difficult times. So with that, I would like to close off and say goodbye.